Thanks very much, Desiree, and thanks for the invitation to present, and thanks everybody for showing up at the meeting. I was certainly thrilled to see the Bahamas was undamaged, at least on this side of things, so glad to be here. As Desiree suggested, she'd asked me to come not so much to give a deep dive into Fountain's scientific approach. I'll touch a few slides on that towards the end of the presentation. But rather, she wanted me to try to bridge the two presentations that you've seen earlier this morning, the scientific presentations that we saw from Dr. Hare and Dr. Hariri, and then sort of the ethical and IRB-based discussions that we had on the panel. And so what I've tried to do in this presentation is bridge that gap and in many ways answer a question that I think we should all be asking. Over the last year, I'm sure everybody in this room has seen the New York Times articles and other articles highlighting some of these high-profile stem cell failures, and particular with the macular degeneration cases. It's very easy to jump on that train, and it's very easy to raise this specter of failure. But I think the important question to be asking is, well, what do we do to resolve that, and what should the models look like to address some of these very real concerns that come out of high-profile failures like that? So the presentation that I'll give today will address at least our thoughts and our position on what can and should be done to address that. So I'll start by saying, even in the traditional drug development pathway, and by that I mean, the Pfizers and the Novartises and the Roches of the world, the model that they're using, which is well accepted worldwide, it's an abysmal failure, not only from a financial perspective, but quite frankly from an efficacy and even safety perspective. We see the overwhelming majority of cells that go, or not cells, of drugs that go through the development pathway fail to go to market. And those that do go to market oftentimes have post-marketing surveillance problems, or again, something like the case with Vioxx, where we see years and years after and millions of patients later that there's a problem associated with the drug. So I think we can reasonably conclude even our accepted kind of FDA framework with historical or traditional drug development, that model is broken. And and as Kathy will tell you, the clinical use of drug models is probably also broken. And I think we can look at some of the challenges with the existing clinical trials model, and I'm not sure that we need to go through these point by point, but I'll simply say universally, we don't have great mechanisms to ensure patient compliance once they take the drug or get the device, or in our case, take the stem cells. Very difficult to retrieve that patient and make sure that they comply with follow-up or other study protocols. And I'm going to really focus a lot on the rest of this presentation on data collection, or perhaps more importantly, the lack thereof. And one of the things that I want everybody to be sensitive to, Josh in his presentation talked about how they use 6-minute walk test as a metric, and that's a well-accepted metric. I think he probably also had some ejection fraction imaging studies. The challenge with those and most of the other clinical endpoints that we look at is that you might follow a patient for 12 months or 18 months or 24 months, and if you're lucky, you'll bring that patient in 3 or 4 or 5 times to do those tests. And when you have relatively small cohorts, typically 30, 40, 50 patients with 2 or 3 clinical endpoints or time points, I think it's very difficult to argue that you can get statistically relevant data coming out of that. In drug studies where your numbers are up high, maybe a little bit easier, but certainly in device studies or in the context of our regenerative medicine studies where these cohorts are typically around 50 patients, I think that's an extraordinarily hard model to drive effective product design in. And finally, I just want to hit on ethics, because this is something that, again, throughout the presentation I'll come back to, and we've talked a lot about that over the last couple of hours. But I want to hit on this from a patient perspective. And as scientists or as physicians, I think in our minds these are very clear studies with very clear rationale on why a patient would enroll. But the reality is, I think if we look objectively, very few patients truly understand the risks and benefits of participating in a clinical trial. And quite frankly, and many times I think their interpretation or their ability to judge that objectively is shifted dramatically by their own medical conditions, right? You have a very different set of optics of evaluating risk and benefit if you have a child with cerebral palsy or a parent with Alzheimer's than you might if you were just objectively looking at it from the outside. Patients also have relatively poor access to study coordinators. Very difficult to report an adverse event once a patient has gone out the door, again, even in a traditional academic study or academic model. So again, we'll try to address some of these challenges throughout the rest of this presentation. If we extend that a little bit to the medical tourism model, and I know that's not maybe the best marketing term, but that's fundamentally what we're talking about. I think almost every of those bullet points that I just highlighted probably get considerably worse in the context, again, of medical tourism. So the last bullet point really is for those of you that have a short attention span or you're ready to go to lunch, I tried to summarize the rest of my presentation here in blue, and that is what proactive steps can we make to change the design or to change the outlook of our clinical studies. And there's really 6 pillars that I'll talk about in the rest of the discussion, and those are forming the underpinnings of the clinical studies that we're trying to do. So first of all, we're trying to look at which patients are the most appropriate for these clinical studies. And our philosophy is that we want to shift to an earlier intervention model. And I'll talk about this again a little bit later, but it makes very little sense to me to try to cure a patient with catastrophic damage. A patient comes in with Alzheimer's, they've got drool dribbling down their chin. It's catastrophic damage accumulated over perhaps a decade or 2 decades. I think it's extraordinarily optimistic to think that we're going to fundamentally shift that model with a single injection of 100 million cells. So can we shift that model to an earlier intervention? Can we use advanced diagnostics and identify those patients in advance and treat earlier? I think for obvious reasons, it makes sense to improve our informed consent process, and I'll go through a few specific recommendations of things that we're trying to implement in our model. Another topic that I think is critical is GMP manufacturing. We all throw about that term, and I hear lots of people talking about their GMP manufacturing facility, and I go into a lot of GMP manufacturing facilities. And the reality is there's not a hard set of rules or standards. There's not a body that's out auditing people for their GMP procedures, and so that gives a lot of leeway for interpretation. I just got back from Mexico a few days, and I spent an hour on the phone with the academic founder of this lab telling me about how they exceeded FDA requirements in their GMP manufacturing facility. I spent 15 minutes looking through the window into their GMP manufacturing facility, and I saw ceiling tiles that were cracked so that the air from the plenum wasn't going through the HEPA filters, it was just going straight into the air. I saw the waste bucket and under the hood overflowing with bleach. There was no particle counters in the hood, and on and on and on. I literally wrote down something like 20 infractions that I could see just looking through the window. So my point here is I'm not trying to advocate, and in fact, I don't think that we need full pharma industry GMP, right? Tracing whether or not you had a blue pen or a black pen in the clean room might be beyond the point of diminishing returns. But I think we can nail down 90% of the risks with 10% of the effort if we do that well. And there needs to be places and mechanisms in place to assure patients that we're actually following those requirements. We've talked about wearables loosely. I'm going to talk quite a bit about wearables in a couple of slides here, but I'll simply say we're at the pivot point of what I consider to be a massive paradigm shift in the way clinical trials are run. Yesterday's 6-minute walk test where you have to come into Dr. Hare's office and walk on a treadmill, is going to be replaced by me pushing a message to the patient saying, we'd like you to do a 6-minute walk test again today for the 340th time in the last 12 months. And I would argue that we get a much more comprehensive and more accurate data collection than you do with that once every 3 months or 6 months or 12-month snapshot. We also historically talk about a single dose and how that single dose is, again, going to reverse heart failure or Alzheimer's or autism or cerebral palsy or whatever it is. Scientifically, I find that very hard to believe, and I'm not quite sure how we got into that mindset. We've been there for 20 years, and I think only recently are we starting to see this concept of serial dosing being applied. And I think that, again, that will profoundly change the impact of stem cell therapeutics. And the final bullet point, and again, I'm stealing most of the thunder for the rest of my talk, but I know everybody's hungry, so this gives everybody a shot to sneak out and skip the rest of my talk. But I find it remarkable that today we live in a community where you can go onto Amazon and find the most remote product, right? 40-weight ball bearings, and get 373,000 reviews on that product and see exactly how it's worked or if it's worked or why it didn't work. And you can see not patient, but let's say consumer experiences with those products. And yet, I would challenge you, other than a few kind of hard to find forums that, you know, I wouldn't say are overly well monitored or modulated, you're very hard pressed to find patients' experience if they've gone to a stem cell clinic, whether it's inside the US or outside the US. And I think, again, we need to modernize that platform. So I'll talk about each of those bullet points in a little bit more depth throughout, but that really forms the core of the presentation and the core of trying to create this bridge, again, between the science and what would look like a more ethical treatment pathway. Before going deeply into some of those, I just want to put some of this into context. So I wanted to take a step back, and I'll just highlight some of the points that I just made in summary there. And say, you know, we've looked now for 2 decades, advertising very well, I'll add. We're great at marketing, if nothing else, in this field. But saying that regenerative medicine and stem cell therapeutics are going to form the next pillar of healthcare. And there have been some remarkable advancements during that time, right? If we look back 20 years ago, the Edmonton Protocol was first reported, and we showed that we could definitively cure diabetes, and we still have patients today from that initial cohort that are insulin-free. I think something like 16 or 18 years is the longest surviving patient right now. That's a phenomenal success. I think we would all agree in the field of immuno-oncology, we've also made some remarkable strides. Orthopedics may be a little bit less dramatic, but quite frankly, probably the most efficacious example of stem cells. But in so many of these other applications where we've heralded these amazing opportunities, I think we can all argue we failed to deliver on the hype and the promise. And so, of course, the question is, why have we failed in that regard? And I think, again, we have to take a step back and look at this from both an ethics perspective and a scientific perspective here. We've certainly had an eager media to listen to our scientific and academic community, and we certainly have set unrealistic expectations. We've also had a habit of promising these results for patients that, again, in my opinion, are probably past the point of no return. We focused on this magic bullet, one single shot, and that's going to take care of everything that ails you. So we've really set the field up, I think, with some of these expectations for failure. And I think, again, we need to take a step back. If we also look at this from a clinical design perspective, the overwhelming majority of stem cell studies that are done today are done with small cohorts of investigator-initiated studies, 30, 40, 50 patients. I think Josh said he had 70, that's a big study. That's an impressive study to be done. And with 3 or 4 data points or 3 or 4 time points, it's a very difficult model for us to figure out what cell type, what delivery mechanism, what timing, what dosage, very, very complex therapies. I think it's difficult for us to really zero in on an efficacious treatment in this kind of model. The other thing I want to point out is this reliance on a single cell type. And it was a great discussion at the end of the last session. And I was actually thrilled to hear Bob Hariri and Josh kind of merge together, and I'm putting words in their mouth, but they effectively said maybe cell type or cell definition doesn't matter so much. And I think that's probably an overstatement of what they actually said. So I'll maybe say it a different way, which is, I think you could probably argue that different cell types might work better for certain problems than others. And so I look at an approach and say, perhaps as we try to start treating patients more proactively, we should have a spread of different cells. And maybe there's one cell that's better for orthopedics. Maybe there's one that's better for neurodegenerative diseases. Maybe there's one that's better for improving cardiovascular health. And again, if we take a step back, most of our dependence on a single cell source is driven by commercial concerns or intellectual property concerns, not what works best for the patient. And as we think about clinical trials design, or in my case, commercial considerations, I think it makes sense to say perhaps we should have multiple cell types in our quiver. If we look at this from an ethical perspective, and I touched on this at the beginning of the presentation, but it is important, I think, to look at some of these high-profile failures, learn from that, and see how we can, again, redesign and step back more successfully. I find it remarkable that in those macular degeneration cases, the investigators didn't even follow their own protocol. Protocol that they had an approval for was for treatment in one eye at a time. They treated both eyes. You know, how that can happen is beyond me. They were using a cell type in the eye, directly injected into the eye, that had essentially no experience with a site-specific injection like that. Again, a remarkable oversight that's very difficult to, in any setting, justify. You may have also seen the cases recently with E. coli contamination. And I talked about this a few slides ago. Most manufacturing sites evolve out of a physician's office or a small academic laboratory, right? Most of them are not evolving out of cell gene therapeutics or some giant pharma model. And the simple reality is, if you've got an academic lab, you probably don't have the resources to have half a dozen people on your quality assurance staff. You certainly don't have the same resources that a cell gene or other large pharma has. So I think, again, it's important for us to try to zero in on a definition of good manufacturing processes that can fit. Again, I don't think that has to look like large pharma, but it should probably be a significant step above some of the laboratories that we're seeing right now. And I certainly think having an audit capability, in fact, I thought about putting a sub-bullet point here. I think this is a fantastic commercial opportunity here, right? Why not set up a company that goes out and does independent audits of these manufacturing facilities? That's probably a lot better way to make money than trying to develop a stem cell cure that works. So there's your commercial tip for the day. So finally, on the ethics, I think it's also important to look at this on a positive note, or maybe end this with a positive note, which is, despite some of these ethical considerations, I find it remarkable that we still predominantly live in a regulatory framework that prevents access to cells for patients that have absolutely no other option. And I think most of you probably recognize these points, but I like to use myocardial infarction or heart failure as the poster child of this. A few years ago, I went back through the literature, and comprehensively looked at all the different meta studies and counted up something like 6,000 or 8,000 patients. So I didn't want to do the work again, so I just rounded up and said it's been about 10,000 patients. So honestly, I don't know if it's 10,000 or 15,000 or maybe 20,000 at most, but it's not a lot in the grand scheme of things for 20 years worth of effort. In that same time span, millions and millions of patients have died from congestive heart failure or other heart-related issues. If you're diagnosed with congestive heart failure, you know exactly what the next 4 or 5 years looks like, and it's terrible, right? You're not going to spontaneously get cured. You're going to get a heart transplant or you're going to die. So I look at that and I say, well, that's a pretty good model to, in Bob's word, be a test pilot, right? We don't have a lot of test pilots, and he was coming at that from the perspective of the scientists, but I also consider that from the perspective of the patient. And I think if we had a room full of people with congestive heart failure in here, probably have a very different lens of risk-benefit than the FDA might, or than we might as scientists. And I think it's important to recognize that. Again, if we go down the rest of the tick list there, whether it's spinal cord injury or autism, there really aren't clinical alternatives. So I think that should change our optics of risk-benefit. So I don't think we want to be, stupid with giving people completely untested things or things that are manufactured in an inappropriate facility. But I wanted to at least end that sort of ethical section by keeping in mind this very serious and realistic snapshot of what would drive a patient into these types of studies. So I won't belabor this because I think I've already kind of hit this point. But I want to highlight again, my perception is that the likelihood of curing a patient with catastrophic damage when you've got billions of cells that are damaged with a single injection of 100 million cells, the vast majority of which are cleared in the first 24 hours in the lung, and most studies will show only 1 or 2% that end up in the infarct area. I find that a relatively optimistic model to say that we would repair that. Now, if we did serial dosing, if we combined that with other rehabilitative therapies, all right, maybe that increases our likelihood for success. That I think is particularly true in the context of spinal cord injury. Even if we are able to make new connections, and we're talking millions of connections, think about how long it takes a baby to learn how to walk. They've got all those connections there, but it takes them years to really learn coordinated walking. And yet, the vast majority of clinical protocols in spinal cord injury squirts them in and starts the clock ticking and hopes that 3 months later they're miraculously going to get out of the wheelchair. There's only one study that I know of that was done by Wise Young out of Rutgers University in China that combined the stem cell injection with 6 months of intensive rehabilitative therapy. 6 days a week, 6 hours a day. And I forget what the exact results are, but I think they had roughly 20 or 30 patients in that study. And these were complete spinal cord injuries. Something like 75% of them walked again. I find that, a staggering study. And again, I sit there and say, why aren't we taking that type of approach in all of our studies to combine not only the stem cells, but these other concepts of rehabilitative therapy to maximize the impact of the intervention? Okay, so despite all of those different grandiose statements, I think the one thing that we can all agree upon in this context is that stem cells are overwhelmingly safe. There have been, of course, some high-profile examples where they weren't, but most of these are pretty easily mitigated, and I think everybody here understands that, so I won't go through that. So I want to transition my last 5 minutes or so here, again, into sort of the positive version and the positive outcomes here. And I'll simply highlight, I think the Bahamas in particular has been a very proactive government in terms of adopting an appropriate risk-benefit study and adopting a more aggressive strategy to give patients access to therapies. And so again, the question then is, as we take these more proactive regulatory stances, what can we do to increase the likelihood of efficacy and safety and improve the ethics? This is just a reminder of the bullet points that I made earlier, and I think I've got one slide on each of those. Which patients should we target? Again, should it be the worst of the worst, the people with catastrophic damage? Very complex to transport these people. I think low probability of success for these complex patients. And again, from an ethics perspective, very difficult to make an objective risk-benefit study. So my feeling, again, is that perhaps our goal should be to slow the onset of chronic disease rather than reverse catastrophic damage. And where all of that leads you, and I think Josh Hare has been a real visionary in this respect, is to this aging and longevity model and trying to shift the care from reactive to proactive. And that's exactly what we believe. And again, I think Josh has been a great visionary in that clinical model. If we look at the informed consent process, there's so many simple things that could be done. Why do we not require all informed consents to have essentially a counterpoint? It's no different than when you go to vote, right? There's a positive statement and a negative statement for any ballot measure that's on the ballot. Why don't we do the same thing with clinical trials? Few patients really have the resources to question the stem cell protocols or to understand them. So again, why isn't there a source where they can objectively go and get information on a spinal cord injury study or an autism study? There are not 10,000 different clinical indications. There's probably 20. Why isn't there an AABB or even an FDA white paper on each of these that can highlight the pros and cons? Cumulatively, I think the message there is that there's essentially no resources to assess the credibility of clinical studies. Clinicaltrials.gov is a complete joke right now because 80% of the studies on there are from Afghanistan or China or who knows what, but not something that went through the FDA or another regulatory body. And they use that as a tool, right? They're trying to get credibility from that. So again, why not require an independent risk-benefit analysis? Why not require the publication of Data Safety Monitoring Board reports? And as I said before, why not enable some sort of patient review mechanism like we see in Amazon reviews? None of those are that difficult to implement. I talked a lot about improving the manufacturing processes. I think maybe those 4 bullet points there, training, environmental monitoring, in-process testing, those could be debated, but I think we would all agree we can probably nail down 3 or 4 pillars of manufacturing that we want to do well, and maybe spend a little bit less resources on determining whether it was a blue pen or a black pen that went into the clean room. The data collection model, I know I'm running short on time, so I won't go overboard here. But I'll simply say the fact that we have access to wearable biosensors will completely change the way medicine is practiced, not only from a telemedicine perspective that we're hearing more and more about in the news, but again, as I made the comparison to a very difficult model that Josh Hare has to bring a patient in to do a 6-minute walk test, we can push that test to the watch, anytime we want. And if you think a few steps ahead of what else can you get from an Apple Watch, I can not only tell the number of steps a patient is taking, I can look at step rate. Gait rate is one of the most highly correlated, age-related metrics that you can have. You can take that a step further, and I can distinguish whether the heart rate was taking place because the patient was exercising or because he was under stress because somebody cut him off on the freeway. All of that you can determine from an Apple Watch. So it's not traditional data that comes out of your Apple Health Kit, but we can see those data points, and with relatively little AI back engine, we can determine these differences. So again, I think this is going to completely revolutionize how we're able to interpret data from patients that we treat and then send off remotely. And then finally, and again, I'm sort of repeating myself here, but trying to treat earlier and trying to treat with repeat dosing, I think, is a critical improvement that we can make to the overall paradigm. And as I said before, also perhaps even using different cell types to provide a broad spectrum approach. We have, again, all of these different community apps, I'll say for lack of a better word, right? And I find it amazing that on Strava, you can see what the pulse rate of your friend was while he was riding his bike up Page Mill Road. You can, look at these incredible reviews on Amazon, or you can go to YouTube and find out how to change the radiator on a, 30-year-old car. All of that you can find out, but yet for a $30,000 stem cell procedure, we're completely blind. That's just, ridiculous, and we should be able to change that. So I'm going to just skip through fountain-specific vision. Bob Hariri talked a lot about this, but and actually Josh's model, I think, also addresses what's the impact of restoring and replenishing a person's stem cell reservoir. And obviously, we think that there's all sorts of positive impacts on delaying the onset of age-related decline. And I'm going to end with a couple of summary points here. And then I want to show a 1-minute video that I think really takes all of this home. And sadly, I didn't make this video. I wish I did. But it's, I think, a great way to end. So I'm just going to skip ahead to that. And hopefully, this will play through. So I'll just let you hear this.
What will your last 10 years look like? Will you be quick enough for a game of tag with your grandchild? Strong enough to embrace every moment? Will you grow old with vitality? Or get old with disease? It's time to decide.
So with that, I'll end. I think that's really the rally call for all of us. I think that the therapeutics that we have on offer can make that difference, and that's certainly what we're targeting at Fountain Therapeutics. So thank you.